All right, welcome everyone. Thanks for coming. We're going to get started with our first MIBI workshop. Just some brief logistics before we get started. For those of you who are on Zoom, all the questions will be you can do through the Q&A tab. On there for people in person, we'll just do normal Q&A at the talks. There's going to be the same Zoom link for all three of the morning sessions. The afternoon sessions will also use their own same link, but it'll be separate since that's not going to be in a seminar. All that information was found in the emails and also is on the website. All the talks today will be recorded and posted after the fact, so if you miss any, no worries. And check the website for any details. All this stuff is also there and after the fact. And with that, I will turn it over to Mike, who will get us started.
Great. So welcome everybody. Really happy to have you all at our inaugural workshop on how to do highly multiplex imaging with MIBI. And to start off, I just wanted to kind of give a little context for how we got to the point of even really caring about doing high dimensional imaging and what sort of problems we wanted to approach with this. And it kind of started, for me a few years ago, and, really trying to understand this idea of how sort of single cell function.
The audio is not coming through.
It's not coming through. Oh, it's muted.
Don't unmute that one.
Unmute
It should be
that.
coming through the room speakers.
Where the drums coming.
We didn't even see maybe can. Thank you. Okay, try talking now.
Can you hear me?
Okay,
Just you can do interpretive dances.
yeah. Okay, sounds good, says Tyler.
Sounds good. Okay. So it was to kind of get at this question of trying to understand the nested organization in tissues, sort of thinking about from the perspective of if we start off with single cells, how are those put together into multicellular structures, and how those multicellular structures put together into sort of functional units in organs and in mammals, and sort of how do these two things feed back on one another. So you have single cells that are sort of under their own control, but they're also influenced by what they're surrounded by. So so this kind of prompted us to really want to get at this question, like how do these two things feed back on one another and to give you sort of a composite functional response, sort of as an emergent property. And so, this has kind of led us to start to think about these things in terms of taking tissues and thinking about them in terms of like a spatial ontology. And so what do I mean by that? So, a spatial ontology for this, for instance, is that we could think about this cartoon as like what types of cells it's comprised of on a bulk basis. And then we could also think sort of, if we think about it, if you just sort of blurred this whole thing and then looked at what the more macroscopic structure of that unit is, we could also think that we could also see sort of from a crude level that this structure that kind of resembles a lot of glandular tissues, has sort of a concentric organization. So there's epithelium on the inside, and there's something that looks like fibroblasts and smooth muscles on the outside. And then lastly, we could also think about this in terms of reoccurring spatial relationships between different cell types. Like, so for instance, the top one says that, by and large, all of the cells the blue cells look kind of like macrophages, are always pretty much very close to the cells, those long spindle cells that look kind of like fibroblasts. And we could also think in terms of relationships like this but in a sort of conditional fashion. So for instance, at the bottom, when you see the cells look more like lymphocytes, whenever those are close to those epithelial cells, we see that those epithelial cells are upregulating some kind of transcription, which we, denoted in red there. So, and so what we've tried to do is, okay, so what we've tried to do is figure out ways to, to generate those data, and then to try to pull out features like this in sort of quantifiable fashion to where we can then look at those and we can start to ask, if we think about all those things I just mentioned as being just different types of rules, it's sort of what is the presence of those rules in tissues from people with different diseases or from normal tissues. And from that, can we then say that we see
It's because we didn't make you practice, Mike.
Well, why isn't it advancing? No. Wait, hang on. Here, I'll just use the arrow. I got it. Okay. That disease 1 and disease 2 seem to have these sort of shared features. And then if we then sort of look at those, and sort of think about this as, this little Venn diagram, these are the shared features, then can we then exploit that information to start to think about, functional mechanisms that they share and diving into sort of the nitty gritty of those? Can that be exploited then to also design different clinical assays? And then lastly, can we use it for drug repurposing? So that's sort of the general framework and, philosophy behind what we wanted to do. And to get there, we developed a type of multiplex IHC, which essentially would allow us to be able to measure all of the properties with an antibody-based assay that you would need to be able to make those kind of determinations. And so that's multiplex IMB imaging. And so essentially, we have tissue on a slide. We can take that tissue and then stain it with a panel of antibodies. Primary antibodies for each one of those antibodies are tagged with a different metal, and each one of those metals has unique mass. And so if you're familiar at all with immunofluorescence, it's really sort of very analogous to that. So, the readout here, instead of being what wavelength of light that the antibody emits light at, it's simply what is the mass of the tag on the antibody. And so by doing that, we can stain and read out around 40 tags simultaneously. And so the way we do that is once the tissue is stained, we put it into a mass spectrometer, into a vacuum chamber. It's got a stage that can move around. We can drive it around and select the field that we want. And then an ion beam is focused down to a small spot size, and it goes sort of pixel by pixel across that field. And as it sits on each one of the pixels, we sputter off material, which includes those mass tags that are on the antibodies. And ultimately, those mass tags are read out by a time of flight mass spec. And we can then take those data and spatially register them and create multiplex images that look a whole lot like what you typically see with like a light-based microscope. And so, when we first started off with this, we were using sort of equipment that wasn't purpose built for it, and we were it's kind of remarkable. I was actually just telling Noah this the other day. It would take forever to generate very, very small fields of data. And so it was no problem, actually. You could manually annotate every field. It wouldn't have mattered. I mean, the fields were tiny. Since we sort of built purpose-built instrumentation, we've now gotten to the problem, a good problem, which is the data scale and complexities at a point where it's now very untenable to be able to do this in a way and be able to do it manually without having automated, robust tools. So, the focus of our labs, Sean and I's lab, has now shifted towards how do we make this then, how do we make sort of multiplex antibody staining and imaging something that can be done in a repeatable fashion, robustly, and then how do we create sort of the downstream analytics to also ease the interpretation of those data. And so, And just to kind of give you a high level kind of overview of some of the stuff we've been working on over the last 2 or 3 years that, a lot of which I think is going to be covered during the workshop. If we start off with the multiplex IHC data by MIBI, we have now I think a pretty robust way to sort of denoise and background subtract these data and do a lot of things that would typically have to be done manually, like image thresholding and signal thresholding. We can then take those data and some really great work that Noah did in collaboration with David Van Balen's lab at Caltech. We now have, really great deep learning tools for whole cell segmentation, and we were able to get human level accuracy on that segmentation data. So this is something, again, that in our first few efforts took a very long time, and now it's something that's really more or less kind of fully automated. Then once we have sort of, the pixel level data and the cell level data, as we'll talk about today, we can kind of look at those at 2 different feature levels, and we can look at those in terms of categorizing cells or categorizing pixels. And so the latter Candace we're going to be talking about today a lot, the great stuff that she's done with pixel clustering. And then lastly, once we have all of that, we can then start to think about these sort of larger organizational features of, like, once you know what kind of cell types we have, how are they put together into sort of microenvironments and neighborhoods, and even do you have like microenvironments comprised of microenvironments? You can kind of see how this could keep going out and scaling up. And so for instance, and what do I mean by that? So here on the left is an image where we've drawn outlines around all the cells in the image, and then the cells have just been colored by what the cell type is. So, maybe it's a T cell or macrophage or something like that. And so, the tools we're going to show you today is essentially being able to take data like this, and then in an automatic way, be able to then understand sort of how are those cells grouped together in regular patterns to form sort of local neighborhoods in the tissue. And so just to give you a quick overview of the workshop, the things that we plan on going through today, over the next 3 days, is we wanted to actually kind of walk through all the stuff that, that we do to put some of this stuff together. So all the way from selecting antibodies, how do we do that? How do you validate them? How do you assemble those antibodies into a multiplex staining panel? Then, also what kind of considerations should you put in when you're trying to design the tissue cohorts? The tissue cohort simply saying, irrespective of what kind of platform you're using, we can't, I think nearly all multiplex systems, we still have to be kind of strategic about what we scan and what depth. This is kind of, the sort of dirty little secret in anatomic pathology is you never scan everything, right? So, and it's sort of, I guess there's kind of this fallacy where people are like, oh, if I scan the full tissue section, it's like, no. So, when you resect out, say, something like a tumor, right? This is usually like a macroscopic object. And when that goes into the path anatomy lab, the techs go through and they're like, oh, I'm gonna take a block here, maybe they'll take like 20 blocks. And each of those blocks in total might be, less than 10% of the total bulk. And then when they look at these tissues, right, you're taking those 2 millimeter thick tissue blocks, you take a recut that's only 4 microns thick, right? So you can see that ultimately, even if you're doing full tissue sections, it's not even a single percent of the total bulk. So for that reason, it's important to remember, and we're going to talk about all this, it's that you will always be selectively sampling with tissue imaging. The question is how do you do it intelligently, and how do you do it in a way that actually is informed by the disease and can get you the answers that you want. Then we're going to go through sort of our pipelines for low-level image processing that we've gotten to a way that we can sort of automate these things now. Then once we've done that, we can show how we can use the output of those pipelines to do pixel and cell-based annotations. And then lastly, once you have all of that, you can start to ask a lot of fun, cool questions about what cells are next to what, how are they put together, how does that affect sort of functional marker expression in different cells, so on and so forth. And and sort of in the process of putting this all together, one of the things we really notice is that really the vast majority of this is sort of platform agnostic, and you can kind of apply to any antibody-based. The stuff that's in green, the low level image processing, this is really the only part that's necessarily sort of MIBI specific. So, you know, I think that, hopefully this will be irrespective of whatever, you know, system that you're using. I think there's a lot of stuff here that will hopefully be good advice and good practices that can be carried over to whatever platform that you're using. And so that kind of gets, so our ultimate goal at the end of the workshop is that, you know, you'll be able to go back, you'll be able to implement the software and the reagent workflows that we talked about here in your own studies. So the sessions then, we've tried, you know, this is our first year doing this, and we've tried to center them around examples from like published studies that we've had or ongoing studies that we have going on now to kind of give it a, you know, a little context so it's not so abstract. Morning sessions are all talks. I know we started early, 8 a.m. and there's a reason. So the reason is just to be, you know, there are people over, there are folks in sort of, you know, all over Australia and Europe who are interested in sort of seeing all this. So we wanted to try to get as best as we could, like hours that, you know, everybody would be awake. Lunch, so this is the most important part of the symposium. There will be no sandwiches. So you know why? Is it because this workshop, I get to pick the food. And so, you know.
Tell them how you feel about sandwiches. Mike.
Yeah, so, you know, so, and just so, and I know what you're all thinking, you're like, oh, what are we gonna eat if we can't have sandwiches? Well, we'll find out. So no sandwiches in my workshop. So, and then afternoon, this is the other thing that we thought about was sort of, is that, you know, we're gonna have a lot of technical talks in the morning. And so presumably, and some of this stuff, a lot of it's gonna involve sort of implementation of software tools and all that kind of stuff. So the way that we've kind of tried to structure it so that you can get some sort of personal attention if you're having trouble implementing some of those, is that every day in the afternoons, we're gonna have what we call office hours, where essentially the material that we've covered either that day or the day before, there's gonna be people available over in person or over Zoom for, you know, 2 or 3 hours to either talk through problems you've had in, you know, previously when you try to do this kind of stuff, or problems that you're running into when you're trying to, you know, install these different software packages. So hopefully that'll be helpful. So, and so in terms of the image analysis software, all of it's freely available. We've tried to put it together in easy to use notebooks. The stuff that is sort of platform agnostic is the ARC analysis platform, and the one that is sort of more specific to MIBI data processing is the talking repository. We're also, in addition, some of the things that, in terms of how to install these packages, we've got a wonderful team of computational scientists that are on staff here, and they've put together some step-by-step video tutorials that will take you through how to put those on your computer if you're having trouble. And then obviously the help during the office hours. So, and I want to highlight for you all the MC, and I spelled it for you, Candace. Yeah, the MC of this workshop, Noah Greenwald. Oh yeah, so he's gonna be here. No, it's not pulsing. So he's Noah's put a lot of effort into this, and he'll be sort of leading the sessions and giving some context to each one of like why why we put them together, and sort of also leading the Q&A. Our workshop presenters, we have technical talks by folks from the Angelo and Angelo Labs. Adam Cagle, who I could not find a picture for him, but because of his huge beard, I thought Simpsons Moses twice. So, and you'll see him. You'll be like, oh yeah, he does make him look like Simpsons. So, then our pre-recorded tutorials, Cami, Shri, and Alex have put a lot of work in all that. They'll also be available during the office hours, I think tomorrow and Thursday, to help take you through personally if you're having any trouble installing any of those. And then we also have some guest speakers, which I think is great. So Eric Abel from IMPAD is going to talk to us actually today or before lunch about what's going to be coming out on the MIBI Scope platform. Barbara Engelhardt, who is coming now at Stanford University. He's going to give a great talk on spatial registration of different types of data sets, and also some really cool machine learning tools for identifying spatial relationships between cells, relating those to sort of expression features. And then we're going to have two folks from WeHi, Marie and Kenta are going to talk about a tool that they've developed for being able to do field selection directly on H&Es and being able to import those into the current MIBI scope system. And so now I know what happened. If you move the thing, you have to go back. Okay, I got it. So just to give you a quick overview day by day, on the first today, we're going to start off with building an antibody panel. It's going to take you through sort of all the stuff around, like we were talking about before, you know, how do you pick antibodies? How do you prioritize which antibodies you should put into a panel? You know, what are the best ways to be able to test these? You know, what's a good, what's the results of a good validation versus a bad validation? How do you, and then again, sort of everything I talked about before in terms of considerations for how to put together a, you know, well-designed study in terms of what tissue you're going to look at and what breadth. Then Eric is going to talk to us about the IMPAT product roadmap right before lunch, and it's med wraps. Oh yeah, that's right. So you know lunch? No, I don't know. Is it med wraps? No, it's not. What is it today? Oh no, I didn't check. All right, so as I put it to the logo.
One of the days it is.
One of the days is med wraps. So. What is it today? What is it today?
Caribbean today.
It's backyard.
Oh, it's backyard. Oh, beautiful. Okay, yeah. So we'll get. All right, you'll see. I have that on the last day, but it's today. So, and so office hours will be with the people that are giving the two sessions: Aaliyah, Ben, and Adisha. On day 2, we're going to talk about the automated processing of MIBI top images. So this is one of those sessions. It's probably going to be more specific to MIBI data. Then we will have a great talk from Barbara. She's going to talk about some of the machine learning tools they've developed for these types of multimodal analyses. Then in session 4, we'll talk about once you've got these process data, how can you start to sort of use different dimension reduction tools at the pixel level to be able to interpret them and pick out you know, meaningful features. And then, is that right?
Yes.
Yes. Then lunch on day 2, start our Indian cuisine. Oh yes, no sandwiches. And then we'll have office hours with a computational team to help you, you know, doing the notebooks. Day 3, we'll start off now thinking about cell-based high-dimensional imaging analysis, and so I think that's when we're going to hit on segmentation. And we'll also go through sort of cell classification and how we do that, how we do cell clustering. And then that's when we'll have our talk from WeHi after that that sort of goes through this new interface they found that if you're using the MIBI scope already, I think could, you know, greatly ease the process of setting up runs. And then here, after that in session 6, we'll go through how do we do spatial analyses to actually look at sort of pairwise relationships between different cell types and really look at tissue structure. Sean will give closing remarks, and backyard today. So yeah, so we gotta switch those logos. But yeah, so exciting stuff on the food front. So and then we'll also have office hours again with our comp team.
And then happy hour at Rose and Crown.
Oh, that's right. I put that down. I'm sorry. Yes, happy hour at Rose and Crown on Thursday when the workshop's over. I think it's at, we said 3:30. 3. Okay, it's on the schedule, isn't it?
It is.
Okay.
There won't be a Zoom session for that one.
It will not be, yeah, that would be a little lame. So, you know, those. So in terms of, you know, I just want to say before we pan it over, I just want to reiterate, as Noah said, all the talks are going to be available online. You know, this is going to be our first year, so I'm sure we're going to have some hiccups, like not being able to change slides and stuff. So
And not knowing what day lunch is on.
and not knowing what days the lunches are on. I had the logos. So you know, please be patient with us on that. you know, the other thing is that, and going through this, we kind of, I think when we put it together, we kind of saw potential for making this an annual thing and sort of broadening the scope to, you know, more to sort of all aspects of, you know, these different assays for spatial biology. So with that in mind, you know, we'd really be eager to hear any feedback you have on how we could improve it, or you know, in terms of what was helpful, what wasn't, and also what you'd like to see us cover in the future. So with that, I'll hand it over.